Direct evidence of autism-like behaviors in early animals is absent, as fossils and simple organisms lack the behavioral complexity to meet human diagnostic criteria. However, we can infer the presence of traits resembling autism based on evolutionary pressures and genetic conservation. Social behavior: early animals like sponges or jellyfish, approximately 600 Maya, lacked complex social structures, so social deficits, a core autism feature, were irrelevant. However, by the Cambrian, social behaviors emerged in some species, for example, schooling in early fish, approximately 450 Maya. Variations in social engagement, like preferring solitude, could have existed in these populations, resembling autistic social withdrawal. In early vertebrates, oxytocin-like systems, linked to social bonding in mammals, began regulating group behaviors. Genetic variations, as seen in modern prairie voles, could have produced individuals with reduced social interaction, potentially adaptive in less social niches. Repetitive behaviors. Repetitive actions, for example stereotypic swimming or feeding, are observed in modern fish and invertebrates, controlled by conserved neural circuits. In early animals, such behaviors could have been advantageous for tasks like foraging or predator avoidance, mirroring autism's repetitive tendencies. For example, repetitive movements in trilobites or early cephalopods might have optimized survival in stable environments, where fixation on patterns was beneficial. Autism's sensory processing differences, for example hyper or hyposensitivity, may have analogues in early animals with heightened sensory responses. For instance, variations in sensory gene expression could have enhanced detection of predators or prey in early fish, providing a survival edge. Evolutionary advantages of autism-like traits. Autism-like traits likely persisted because they offered adaptive benefits in certain contexts, even in early animals. Specialization. In modern mammals, autism-like focus on patterns, for example systemizing, aids tasks like tool use or foraging. In early vertebrates, individuals with intense focus on environmental cues, for example currents, prey movement, may have excelled in specific roles, enhancing group survival. Neurodiversity. Genetic variations in neural function likely created a range of behaviors in early animal populations, allowing flexibility in changing environments. For example, solitary individuals might have explored new territories, while social ones maintained group cohesion. Sensory acuity. Heightened sensory processing could have improved survival in predator-heavy Cambrian seas, where detecting subtle environmental changes was critical. Challenges in identifying autism in early animals. Lack of behavioral evidence. Fossils provide no direct insight into behavior or cognition, and early animals' simple nervous systems, if present, limit comparisons to human autism. Human-centric diagnosis. Autism's diagnostic criteria, for example social communication deficits, rely on human-specific traits like language, which even modern mammals lack. Applying these to early animals is speculative. Complexity of autism. Autism arises from complex gene interactions, epistasis, and environmental factors, which are hard to model in ancient species with simpler genomes. Evidence from modern analog studies of modern animals provide clues about early evolution. Fish, zebrafish with autism-related gene mutations, for example, SHANK3, show altered social behaviors and repetitive movements, suggesting such traits could have existed in early vertebrates. Invertebrates. Fruit flies with mutated autism-linked genes exhibit atypical sensory responses, indicating these genes had roles in early nervous systems, approximately 600 Maya. Mammals. As noted previously, mammals like rodents and primates show autism-like behaviors when genes are altered, supporting the idea that these genetic mechanisms were present in their early ancestors, approximately 200 Maya. Conclusion. Autism as a human diagnosis did not exist in early animal evolution, but the genetic and neurological foundations for autism-like traits likely emerged with the earliest nervous systems, approximately 600 Maya, and became more pronounced in vertebrates, approximately 500 Maya. Genes linked to autism, conserved across animals, suggest variations in sensory processing, repetitive behaviors, and social engagement could have existed in early animals, offering adaptive advantages like enhanced focus or sensory acuity. These traits, while not autism in the human sense, likely contributed to neurodiversity in ancient populations, aiding survival in diverse ecological niches. Direct evidence is limited by the simplicity of early animals and the human-centric nature of autism, but modern analogues in fish, invertebrates, and mammals support the presence of these traits in evolutionary history. If you'd like me to focus on a specific period, for example Cambrian early vertebrates, or explore particular species or genes, let me know. Autism in other mammals. Autism spectrum disorder, ASD, is a complex neurodevelopmental condition defined in humans by differences in social communication, sensory processing, and repetitive behaviors. Its study in other mammals is limited because autism is diagnosed through behavioral and cognitive criteria specific to human contexts, and there is no direct equivalent in veterinary or animal science. However, research on mammals, particularly in animal models like rodents and primates, suggests autism-like behaviors and neurobiological traits can be observed or induced, providing insights into potential parallels. Below is an overview based on current knowledge. Evidence of autism-like traits in mammals. Natural observations, non-human primates. Some studies report autism-like behaviors in wild and captive primates, such as repetitive movements, for example rocking, reduced social engagement, or heightened sensory sensitivities. For example, a 2016 study on rhesus macaques identified individuals with low sociability and repetitive behaviors, resembling ASD traits. These behaviors may reflect natural neurodiversity, as genetic studies show autism-associated genes, for example SHANK3, MECP2, are conserved across mammals, predating human evolution. Other mammals. In dolphins, some individuals exhibit atypical social behaviors or repetitive actions, for example circling, but these are not formally diagnosed as autism due to diagnostic limitations. Similarly, anecdotal reports of dogs or cats with repetitive behaviors, for example tail chasing, excessive grooming, or social withdrawal exist, but these are often attributed to stress, neurological issues, or breed-specific traits rather than autism. Animal models of autism. Rodents, mice, and rats are widely used to model autism-like behaviors. Genetic mutations linked to human ASD, for example SHANK3, FMR1, or NLGN3 genes, are introduced, resulting in behaviors like reduced social interaction, repetitive grooming, or altered vocalizations. For instance, BTBR mice naturally exhibit low sociability and repetitive behaviors, making them a standard ASD model. These models suggest shared neurobiological mechanisms across mammals. Primates. Rhesus monkeys with induced mutations, for example SHANK3 knockouts, show social deficits and repetitive behaviors, reinforcing genetic parallels to human autism. Prenatal exposure to valproic acid in primates also induces autism-like traits, mimicking human environmental risk factors. Other species, less studied, but some research explores autism-like traits in dogs given their social cognition. Canine compulsive disorder, CCD, in breeds like Dobermans, for example tail chasing, shares genetic and behavioral similarities with human ASD, particularly in dopamine and serotonin pathways. Neurobiological similarities. Autism-linked genes are evolutionarily conserved across mammals, regulating brain development, synaptic function, and sensory processing. For example, oxytocin receptor variations implicated in human ASD social deficits affect social bonding in prairie voles. Brain structure differences like altered amygdala or prefrontal cortex connectivity in human ASD are replicated in rodent models, suggesting mammals share neural substrates that could manifest as autism-like traits. Challenges in identifying autism in mammals. Diagnostic barriers. Autism is a human-specific diagnosis relying on language-based criteria, for example communication deficits, which cannot be directly applied to animals. Behavioral parallels are speculative and often conflated with other conditions like anxiety or neurological disorders. Contextual differences. Social and sensory behaviors vary widely across species. What appears autistic in a dog, for example avoiding eye contact, may be normal for its species or environment. Research bias. Most studies focus on induced models rather than natural populations, limiting evidence of spontaneous autism-like conditions in wild mammals. Evolutionary perspective. The presence of autism-associated genes in mammals suggests these traits evolved to enhance survival in specific contexts. For example, heightened sensory focus or repetitive behaviors could aid foraging or predator avoidance in ancestral mammals, similar to proposed benefits in early humans. Social deficits might be less detrimental in solitary or less socially complex species, allowing such traits to persist. Conclusion. While autism as a human diagnosis doesn't directly apply to other mammals, autism-like behaviors and genetic underpinnings are observed in species like primates, rodents, and dogs, particularly in research models. These suggest shared neurobiological mechanisms across mammals, rooted in evolutionary conservation of genes regulating brain function. Natural occurrences are harder to confirm due to diagnostic and contextual challenges, but neurodiversity likely exists across mammalian species to varying degrees. If you'd like me to explore specific species or genetic studies further, let me know.